So today it is my pleasure to introduce our speaker, Dr. James Morris. Let's see, I am recording and I'm streaming. Okay, Dr. Morris is a distinguished professor emeritus in biological and marine sciences at the University of South Carolina. He's former director of the Baruch Institute for Marine and Coastal Sciences and a fellow of the American Association for the Advancement of Sciences. He holds a degree in environmental sciences, both his bachelor's in biology, an MA at Yale, and forestry and environmental studies, a PhD at Yale, followed by a postdoc in marine biology at Woods Hole. And he's authored more than 120 publications, a long history of NSF funding, and honors including the USC Research Foundation Award, the Society of Wetland Sciences Merit Award, and his discovery of the stabilizing feedback between salt marsh primary productivity, accretion, and sea level rise really motivated most of the current and ongoing work on basically the stability of marshes under accelerating sea level rise. So it's really been fundamental to what we understand about salt marsh ecology. We're very pleased to have you, Dr. Morris. The floor is yours.
Okay, thank you, David. Thanks for that nice introduction. And I'll start with a little story first. When I was a graduate student in residence at Yale, H.T. Odom was already an icon, and he was a science hero of mine already at that time. And fellow graduate students and I would climb up in the tower of Osborne Memorial Lab. Some of you may know it. It looks like a castle. And there's a tower, and it was rumored that H.T.'s office was in this tower. Probably not true, but we would climb up in that tower and try to imagine what things were like during the heyday of H.T. or G. Evelyn Hutchinson, who I believe was H.T.'s mentor. So I've admired H.T. for forever, and so I'm honored to be able to give a seminar.
Thanks, Jim. We appreciate that. The long history of connecting the center and the fundamental ideas of ecology. And we have some compatriots of HT here in the room, at least one. I think I see Dr. Ewell.
Oh, so. Yeah. Hi, Kathy. Okay, so I'm going to talk about the work I've mostly done at North Inlet in South Carolina for quite a long time. And this is a colonial era map of North, the location of North Inlet on the left. And this
sorry, Dr. Morris, you have to share your screen again. I disabled your share for the intro.
Okay. Where did that go? Oh dear. Close that. Oh shoot. I've lost it. Hang on. Where did it go? Here, here we go. What's that? Oh, yep. Share screen. Okay, there we go. Back.
All right, thanks. Sorry about that.
That's okay. Okay, so here we are. This is North Inlet. From the air on the right, and it's a clean, pristine estuary, and we think that the processes we study here are a pretty good proxy or pretty good sentinel site for climate change studies because it's not impacted by human activities to any great extent. And so I started working here in oh God, a long time ago. Oops. And when I started, the hot topic in salt marshes was regulation of primary production, and it still is to some extent. But at the time, everybody was focused on nitrogen as the important control, and you know, that's a no-brainer, and that's accepted fact now. And here's just an example. This is some post that's in the marsh, and it's is this thing in the way? Yeah, it is in a way. So maybe. Okay, so this, ospreys and egrets land on this thing and they defecate and fertilize the marsh. So it's nitrogen limited. So, but then the attention shifted to sulfide. Everybody was thinking, well, sulfides are toxic. Um, there's something to that. And so I started an experiment with Gary King. Gary King was the sulfate reduction guy back in those days, one of them. And we designed an experiment to precipitate the sulfides in the soil. And we had to do this by adding an iron mineral called goethite. And you can't add a lot of this stuff. So we had these small plots that we set up. Some were fertilized with nitrogen, some with phosphorus, some with goethite. And the idea was to see if we could remove the sulfides, could we increase the primary production? That experiment was really a colossal failure in some ways, because the iron we added just precipitated the phosphate. It precipitated the phosphate as well as the sulfide, and so it created a phosphorus deficiency. But anyway, because we had these fixed plots, I had to develop a way to measure primary production non-destructively. And so we set up these permanent plots, and they're still there today, and we're still doing this. And on these permanent plots, we tagged the stems with bird bands, and you can see these bird bands here, and we would measure the stem heights, and then we would convert the stem heights into weight based on regressions from destructive harvests. And we do this on a monthly basis, and so we can get growth rates, death rates, survivorship. There's all kinds of information on a population level from these kinds of data. And so I started doing this, and within 3 years, I saw just a huge amount of change in primary production. If you look at this graph on the bottom, so here's year one, it was almost 500, then almost 1,000, and then back. And that really just, I thought that was crazy, because aren't salt marshes really predictable habitats? Pick up a tide chart. I was really intrigued by this. And an oceanographer friend of mine, Bjorn Sharvis, said, well, did you look at sea level? And no, why would I do that? And he said, well, it bounces around a lot, especially seasonally and from year to year. And so the figure on the top is a plot of monthly mean sea level, and then the open circles are the sea level during the summer growing season, and that's critical. That's a critical time of year for these plants. And sure enough, there was a correlation there. And so I reasoned that the elevation of these marshes was actually too high. They were super optimal, and more flooding increased production. And the explanation is that the deficit in flooding created or led to high salinities, which decreased the growth of these things. And so, and we see the same thing in other estuaries. This is a plot of primary production of Spartina in Plum Island in Massachusetts. And these are the same plots over time, but plotted against the mean high water level during the growing season. So it's the same plot, but you can see the same thing. High sea level stimulates production. Well, that really led to this model, the Marsh Equilibrium Model, that and the fertilization response we saw. So when I got the idea that, well, okay, if you look at this, there was actually a significant trend, increasing sea level, or increasing productivity over time. And so that made me think, well, okay, these marshes they must not be keeping up with sea level. If more flooding is good and production is going up, maybe they're not keeping up with sea level. So I started measuring the elevation of the marshes with these sediment erosion tables. This is my student PB who's making these measurements here. And I set up these fertilized plots. So one side of this is a control, one side is fertilized. And I was thinking that fertilization would actually decrease the elevation of these marshes. That was the original hypothesis, because you know, everybody was thinking that fertilization increases decomposition, decreases root production, yada, yada, yada. That's not what happened. Just the opposite. So the graph here on the bottom shows a plot of the elevations of these sites. These are controls here. The black dots and the open circles are the fertilized sites. I mean, the fertilized sites, the elevations just took off. And today they're so high that when the tide comes in, it surrounds these little islands of fertilized marshes. So, you know, that was an epiphany. I got it. Okay, that's how these marshes are keeping up with sea level. When the elevation of the marsh is high and when sea level rises, that stimulates growth, which increases the accretion rate. And so the marshes keep up with sea level that way. And there's an equilibrium that's established. So that's really what led to the Marsh Equilibrium Model. And this is a plot now of more of the same kind of data. And the red line here is a MIM calculated elevation. These are elevations over time. The controls are on the left here, and the actual observations are these squares in the, I guess, the end of season or end of year elevations are the green dots. Anyway, the slope here is, you know, about 1 millimeter a year. So the original hypothesis that these marshes are not keeping up with sea level was true. But when you fertilize them, during the period in which we fertilized these sites with the SATs, we're getting almost 5 millimeters a year. So these marshes were growing vertically faster than the rate of sea levels. Then I lost my funding from USGS, and it pissed me off. And so I stopped fertilizing, but we kept measuring the elevations, and look what happened. As soon as we stopped fertilizing, the increase stopped. We are now fertilizing again because I want to see how high I can get these things to go, and I want to see what happens in other ways. So when production increases, sedimentation increases. As you can see here, these plants literally trap sediment, and you know, the greater the density of plants, the more sediment. But the most important thing really is the roots. You know, people even today think that when you fertilize, it decreases the production of roots. Not true. So these are CAT scans made by a colleague of mine, Kathy Wiggin and Earl Davey. And these were taken from these plots at North Inlet. And the upper left here shows phosphorus treated and controls here on the bottom, no difference. And on the right are, this thing's in the way. The right shows nitrogen and phosphorus here and just nitrogen. And these CAT scans, this is an interesting new technology. This is really pretty cool. They spirit these things inside a hospital, literally in the middle of the night. And I'm not kidding. And they run these things through this you know, $1 million CAT scan machine in a hospital to get these data. So the original Marsh Equilibrium Model was was abstract, and it's kind of like a late Picasso. You know, you can kind of recognize what it is, and it's really pretty simple. It just says that the change in elevation, the rate of change in elevation, is the depth of the surface, the marsh surface below mean high water. Okay, here's a cartoon of the geometry, the idealized geometry. So Z is the elevation of the marsh surface, and you know, D is the depth. So it's D times Q. Q is sort of like a settling velocity term, and then KB is the contribution of biomass. But the interesting thing here is that the biomass itself is a function of depth, and it looks like it resembles a parabola. So there's a depth that's too great, where hypoxia kills the plants, and there's a depth that's too shallow, where osmotic stress gets them, and then there's a sweet spot. Well, if you substitute this in for B here, this boils down to a cubic function here, shown here, and you can solve this. And solving it leads to some really interesting generalities. And what I've done here is I've plotted the equilibrium depth. Remember, there's an equilibrium that's dependent on the rate of sea level rise. And so here's the equilibrium depth versus the rate of sea level rise. And there is a side to this that is stable, and there's a side that is unstable. So, you know, think of it this way. If the rate of sea level rise is 0.5 centimeter a year and increases, that increases the depth. That increases the equilibrium depth, but it's stable until you go, well, on the, and the limit here was, you know, 1.5 centimeters or less. And then you can calculate then also what the productivity is. The productivity is a function of depth, and there's a maximum productivity that is a function of, or there's a rate of productivity. The rate of productivity is a function of the rate of sea level rise, because this all, you know, it controls the elevation of the marsh, which controls, you know, okay. So the equilibrium depends on the rate of sea level rise, which you see here in B, and in A, consequently, the productivity, I already said that, depends on the rate of sea level rise. And there's an optimum rate of sea level rise for primary production, and therefore for carbon sequestration. And that's a pretty neat idea. So since then, we've been trying to measure the shape of this parabola using experimental approaches, bioassays, which people call marsh organs. The term was actually coined by Denise Reed, who, when I was explaining to her what this thing looks like, she said, "Oh, it's a marsh organ." So it stuck, and that's what people call it today. So this one here is in Massachusetts. This one here is in South Carolina. And by incubating or growing these plants in these tubes, and the tubes extend all the way to the surface of the marsh, and they're open at the bottom, we can get pretty nice looking data that look like this, which, and one interesting thing about this is that the x-intercepts here are proportional to the tide range. So if you have a marsh that's microtidal, this thing is very compressed, and we have data on that as well. And we've had some fun with this. Here's Karen Sundberg, my technician. She's the winner of the first North American marsh organ race in 2006. Here she is paddling along, putting this thing in position, and it's taken off. People all over the world now are using this. The Dutch have gone crazy. This is a marsh organ of their invention that's out in the middle of the Baltic, where it's got to be pretty robust. And sure enough, the Dutch, they don't, when they do something, they go all out, man. This is, you know, major engineering. Here's one in China. They're using bleachers. This is a macrotitle site here, and so it's hard to use the original design in an environment like that. So they go to bleachers, and they're also looking at competition between mangroves and Spartina. So another word about the nitrogen effect. So we've been dosing some of these marsh organs with nitrogen. This is a great way to measure root and rhizome production, by the way, because, you know, when we start these things, we try to start it with clean sediment that we take out of the creeks. And so there's very little organic matter in it. And so when you grow these things over a period of time, the organic mass that you harvest is whatever was produced since you planted these things. And we've been experimenting with these things by keeping them going for a period of years. And this is the mass of roots and rhizomes that were pulled out of a fertilized organ. And after 3 years, they are basically impacted. But what we've learned from this is pretty interesting. The rhizomes have indeterminate growth. They do not turn over every year the way roots most likely do. These things live on. They live for years, and they just grow and grow and grow. And here in this graph on the right shows the partitioning that we have found between the parts of, you know, the roots and the rhizomes, dead, live, and so on. The rhizomes really are the star of the show in terms of creating long-lasting volume. The rhizomes are just that's where all the action is. And here is another, here are some more data. So we let the controls go for 5 years because they're less robust and they don't fill up the pots the way the fertilized plants are. So this is total plant biomass, live and dead. And so here's the fertilized sites, or organs, fertilized organs. 8,000 grams per meter square is what we're getting out of these things. And here are the controls. The controls have basically reached an asymptote after a couple of years. And then this is total below ground biomass, living and dead. And here are the fertilized pots, and here are the controls. And this is just some more information here on the bottom about the partitioning. So the below ground total in the fertilized, well, in all of them, I mean, you can see how important the below ground biomass is. It's quite amazing. And the annualized growth rates are spectacular. And we can calculate this as well from these curves in these different treatments. And the growth of rhizomes is just amazing. I mean, it really is. So that's neat. And so here's what it looks like when you plot above and below ground biomass versus the elevation or the depth. I'm using dimensionless depth, where the absolute depth is divided by the tidal amplitude, I think, is what I divide by. And anyway, you get these pretty nice, reasonably nice looking curves. So there's your parabola, and both above and below ground biomass basically follow the same kind of model. So we can calculate mass sedimentation rates, and we can calculate the mass of root production and rhizome production and so on. But to convert that into volume, which we want to do because we want to be able to convert that into vertical accretion rates, we've got to calculate the bulk density. And this is the result of a study I did a few years ago. These are over 5,000 samples taken from marshes all over the US along the coast. These are the sites up here. And they follow this self-mixing or ideal mixing model. And the conclusion here is that the bulk density is the sum of the parts, and the parts being the organic part and the non-organic part. And that fits. So here are the self-packing densities here. These are the values. And so we take now, we can divide the mass inputs by these self-packing densities and then get the volumes out. Now, I'll take you through a little example of how this system works. So let's start with one of these parabolas, okay? And so this is going to be the refractory biomass production, which is, below ground biomass. It's the lignin fraction of the below ground production. So, hypothetically, let's say that, the absolute value is not important, but the shapes are. So it's got a shape that looks like this as a function of depth below mean high water. So you divide that, and we're just going to focus for a minute on the organic part. Okay, so we divide that by the self-packing density, and then we can calculate the vertical accretion that looks like this. Okay, now again, forget about the mineral part. We're just focusing on the organic. So if you've got a curve like this, you've got the accretion rate and you've got depth. Where's the equilibrium when the rate of sea level rise is 0.4 millimeters a year? Well, it's going to be, there are two. There's going to be one here and there's going to be one there. Those are the equilibriums. And remember, one is stable and one is not. So this is high marsh on this side. This is low marsh on this side. So high marsh is low depth. Low marsh is high depth. So what happens if the depth is 75 below mean high water? So if we have a point here, well, in this case, the accretion rate is greater than the rate of sea level rise, which we said was 0.4 millimeters. So in that case, what happens? Well, if the accretion rate is greater than the rate of sea level rise, then that has to decrease the depth, right? i.e. increase the elevation. And so that point goes there, boom, and it hits that equilibrium point there. So the marsh rises until it hits that equilibrium. Now, what if we put it at minus 65 over here? If we put a point there, well, the accretion rate is less than the rate of sea level rise, so depth increases. Boom, it goes there and hits that equilibrium point. If we put it at 80 over here, well, here the rate of accretion is less than the rate of sea level rise. The depth increases, boom, and marsh goes extinct, okay? So this point, this is a tipping point, and it's kind of interesting when you think about this, because these points are actually, they're functions of the rate of sea level rise. If you raise the rate of sea level rise, these points move together like that, and then the marsh becomes probably less resilient to sea level rise, future sea level rise. Now, what happens if you add mineral sediment to the mix? Well, you get a curve. Here's the organic part, and then here's the mineral part. So the mineral input increases more or less linearly with depth. And so here's the mineral part. And then you have, in this case, you have two stable equilibrium points. You've got one here, as in the previous example. You've got an unstable point here, as in the previous example, but then you've got another stable point over here. So here you have alternative stable states. This is mudflat on this side, and this is marsh on this side. So for any depth where relative accretion rate is negative, depth will increase. So if we're down here in this region here, the depth will increase. Boom, when you hit that point there. And for any depth where relative accretion rate is positive, depth will decrease. So if you're in this region here, the marsh migrates in this direction. If you're here, well, that's the stable equilibrium point. So that's really how the Marsh Equilibrium Model works. I call this Marsh Equilibrium Theory. And now I want to change gears a little bit. Yeah, okay, there's plenty of time. I'm probably going too fast. So the whole thing that got this started in the first place was the observation that production was a function of relative sea level, which varies a lot from year to year. And so there are these interannual variations and there are the seasonal variations. The seasonal variation, by the way, is caused by the heating and contraction of the ocean. It heats up in the summertime, water level rises on the average by about 30 centimeters. And man, that's huge. And, 20 years ago, me being a biologist and not knowing a lot about physical oceanography, that was a revelation. I had no idea. But there's some other interesting cycles too. There's some long period cycles that sort of makes you wonder, well, are the marshes responding to some of these longer term cycles, like what's known here as the lunar nodal cycle. The lunar nodal cycle is one that's 18.6 years. And plotted here are monthly mean tidal amplitudes from Charleston. And there it is. So boom. Now this is amplitude. And the question is that enough of a change in hydro period to affect the productivity? Incidentally, these times here, these are the times of king tides. You've probably, hopefully heard about king tides. And people, places like Charleston have major nuisance flooding during this part of the tide cycle. King tides. Okay, so, well, look at that, would you? So this is the productivity that I've measured in North Inlet. And there it is, man. That's the 18.6 year cycle. And you can see it in the productivity data. Now, there's a lot of variability for sure, because there's a heck of a lot going on that affects productivity from one year to the next, including these seasonal cycles. And the seasonal cycles vary because, that's a climate-driven thing. That's not an astronomically forced cycle. That's climate, that's heat. That's the position and strength of the Bermuda High that pushes water up against the coast. I mean, there are all kinds of things that affect that cycle. And so there's a lot of variability from year to year. But embedded in that is this long period cycle that you can see here. And it's statistically significant. And now we've also been monitoring pore water chemistry in these marshes on a monthly basis forever. You know, we're gluttons for punishment, I guess. And I've devised a neat little system for doing this using wells that are screened, and the wells are placed permanently in the marsh, and then there's an insert pipe. You can see this insert pipe here. Here's the insert pipe, and it's hollowed out to hold these scintillation vials that are screened with Nitex membrane. And we pop them in there, and they sit in these wells for a month, equilibrating, and then we pull them, and then we put another clean set of vials in when we harvest these things. We've been doing this for quite a number of years, and we go down to a meter. So we do 10, 25, 50, 75, and 100 centimeters. So we have depth profiles, and, you know, you name it. And so what do we see in this pore water data? Well, this is interesting too. This is what the raw data look like. These, and I'm going to show phosphate here because phosphate is the most, it's the most well-behaved. We do phosphate, sulfide, salinity. Well, salinity is well-behaved too. And we do ammonium. And, you know, for sure, there's a cycle here that you can see. Now, this is a seasonal cycle. And the peaks occur during the summer. They're basically coincident. They're coincident with the times of maximum production, which is kind of interesting. That salt marsh does not work like a lake. It's not like you have a phytoplankton bloom and boom, the nutrients crash. No, no, no. Everything here is coincident. So there's this annual cycle, and but you know, we can also pick out an 18.6 year cycle in these data, and that's what the 18.6 year cycle looks like, the curve on the bottom, and that's significant. Now, if we combine these, we get something that looks like that. Now, that's much more satisfying. That looks a lot better. But gosh, there's more here than that even. The amplitudes also change on an 18.6 year cycle. And if we throw in the change in the amplitude, we get this. And this is about the best I can do, you know, pulling the signal out of the noise. This is about the best I've been able to do, but I'm pretty happy with this. Now, and there it is again with more recent data. I'm just jumping, I'm doing somersaults over those data. That's pretty cool. Now, you can do something really interesting with these data. You can ask questions like how they covary. So here's ammonium concentration versus sulfide concentration in the pore water. And this is one annual cycle. And so it's going up, it's going up. And if you look at the whole time series, it does this. It goes up and then it comes back down. Whoa, that's pretty cool. These are limit cycles. Now, this is the symphony part. I don't know if the sound is going to play, but I've set this. I'm going to show you an animation, and I've set it to music. And we'll see. I mean, this looks like this is ammonium versus phosphate versus sulfide, and color is the fourth dimension. Color is the monthly growth rates of these plants. And it looks like a basket, doesn't it? Not quite. All right, let's see if I can get this thing to play. There we go. Okay, so boom, boom. That's one annual cycle. It's winding around. Notice that when the plants are hot, everything's going up. And when the plants are cold, everything's coming down. Remember, I said everything is coincident. And here, when we rotate it, you'll see that it's not really a basket. It's, if it is, somebody stepped on it. You know, it's kind of flat. All right, that's enough. Okay, now, if you've been paying attention, you might be wondering, well, what do the fertilized plots look like? Well, let me show you the fertilized plots. The fertilized plots are chaotic. That's here's the fertilized plots. Yeah, so this is real chaos. You know, there is, they're deterministic, but they're unpredictable. So that's what fertilizing has done. Now, what's really, where did we go? What's intriguing about this is that if you look at the concentrations of ammonium, especially ammonium and phosphate, they are not so very different, are they, between the controls and the fertilized sites? Even though we're adding nutrients, several, you know, we do this periodically during the year. We add nutrients. We put them in the ground, actually, and you know, there's no doubt that some of this chaos is because of the nutrients we're adding. But both of these sites tend to maintain similar nutrient concentrations, which is kind of interesting. Now, the coincidence, I think, is orchestrated by the plants themselves. These plants release a lot of organic matter, and this is a photo of a rhizome and some roots taken through the wall of a mesocosm. And the sediments here were made artificially. There was no organic matter. They were all, you know, the original color was just solid tan, very clean. And within the first summer, they started to turn black. Now, the black color is iron monosulfide, which precipitates. The sulfide is coming from organic matter that's being released by these roots and rhizomes. And the environment here, the microenvironment, is really complex and really kind of interesting. And you see an oxidized layer right around this rhizome here. And here's even a little oxidized layer around this tiny little root that's coming off here. And you can see iron precipitate on the roots here. So this is Fe3 that's precipitating on the roots. And so these plants basically are fertilizing these sulfate reducers by releasing organic matter. Now, we know that there's a lot of acetate in the soil associated with Spartina. That's been established for a long time. These plants are putting out acetate. Now, that may be a product of fermentation in the root system. It would be a less toxic end product than ethanol. And so, maybe that's why they're making so much acetate, but they're making a lot of acetate. And acetate happens to be a perfect substrate for sulfate reducing bacteria. Now, isn't that interesting? Sulfide, which has long been thought to be, a horrendous toxin, is actually, the plants are actually producing this stuff. And that is really, really interesting. And here's a graph that shows the concentration of DOC in the pore water. This is just total DOC, but probably a lot of this is acetate. And it cycles seasonally, and it's high when the plant biomass is high and when the plants are growing. So here's the breakdown of what's in this stuff. We've got a lot of acetate, but we also have a lot of formate in here. But this one especially is a good substrate. Now, here's something else to think about. This is pretty cool. These are cores pulled from those marsh organs, and it's very common that we see elemental sulfur deposits in these roots. Now, the elemental sulfur has got to be coming from sulfide oxidizing bacteria. Okay, so there are bacteria in here that are getting energy from that sulfide, just like the organisms that grow thermal vent communities at the bottom of the ocean. These guys are just getting energy out of the sulfide that they are oxidizing. But a lot of the roots and rhizomes are colored with purple, and there are non-photosynthetic purple bacteria of genera with nitrogen metabolism. Rhizobium or Nitrobacter are two groups. They fix nitrogen and they use sulfide as an energy source. Now, that's really intriguing. So, maybe there's this symbiotic relationship between these plants and those bacteria, and it involves the sulfur cycle. Basically, these plants are harvesting energy and probably nitrogen as well from these bacteria. Now, it's possible that this color is also just a plant pigment, anthocyanin, which would be not so interesting. And we just haven't gotten to the point yet where we know that.
Jim, we've just got a few minutes before. Oh, there we go. All right.
Okay. Good. So these are the conclusions. Yeah. Salt marshes possess stabilizing feedbacks. At low rates of sea level rise and or high relative elevation, plants respond positively to an increase in sea level, increasing sediment trapping and soil biovolume raising elevation. Organic production and mineral sedimentation are both important. Minerals more so at higher rates of relative sea level rise and low elevation, and organic production more so at low relative sea level rise and high elevation. Nutrient enrichment increases biovolume production, and the plants appear to stimulate the production and availability of essential macronutrients to meet the demands, but still they are nitrogen limited. There may be important symbiotic relationships between the plants and microbes, maybe even endosymbionts. Maybe cable bacteria, which transfer electrons through cables, basically. So these plants might be harvesting energy. So the music, She Blinded Me with Science by Thomas Dolby. Some of you are probably old enough to remember this one. And Book It by the Tyson Naylor Trio. And this this work has been going on for a very long time, and I would not have been able to do this without especially the work of some amazing technicians, a succession of technicians. The first was Betsy Haskin here. She's the daughter of Hal Haskin, the Rutgers shellfish guy. And Karen Sundberg is the current, she's the winner of the North American organ race, but she's remarkable. All of these people are, but especially Karen. Karen is just crazy cool. And that's the end. So,
thanks, Jim. Thanks so much. It's amazing to see what long-term dedicated collection can come up with, even if it wasn't in your mind at the beginning. It's really an amazing seminar, and to see it all together, animated and with a soundtrack. It's awesome. So thanks so much. We'll open it up to questions for anyone in the Zoom room. And sometimes it takes a little bit to get the crowd warmed up. So I'll start with one, which is that you know, from all the Marsh Oregon work and racing, you did a lot of work to extract parameters about growth rates, and I wonder if there's any if you think there's any sort of residual bias or effect of the experimental setup in thinking about above ground, below ground biomass, sort of from which it was the parameters like B and et cetera were derived. Has there been work to look at that?
Yeah, it's a good question. And a lot of people ask that, are there, like chamber effects? Yeah. I don't know. It's a good question. And I think the answer is probably yes. Are they a perfect model of a real salt marsh? No, because they don't allow, for one thing, they don't allow for horizontal flow. So if there's like Darcy law flow, there's Darcy law flow in a real salt marsh, but not in these marsh organs. The flow is vertical. It's not restricted, but it's vertical. So that's one difference. So yeah, there are artifacts, I think, for sure.
I see Kathy has her hand up. Kathy, you want to go next? And then we got a question from Kathy Todd Brown.
Unmute,
Yep,
Kathy.
I got it. Jim, I'm wondering about the groundwater hydrology and impacts of runoff groundwater coming into the salt marsh and perhaps affecting nutrient concentrations.
Yeah, that's another good question. Related, because there's no horizontal transport in these little mesocosms. So yeah, that is potentially, I mean, there are probably a number of variables that are important in a real salt marsh that are not important or less important in these mesocosms. So, I'm not, for example, I don't think that these mesocosms are a really good model of growth in the high marsh versus growth on the creek banks, because the creek banks really do get a lot of horizontal transport drainage that affects the growth of those plants. And so I can't model this, but I would posit that these things are probably a reasonably good representation of what drives production on the marsh platform itself. So if you could take the whole marsh platform and raise it and lower it, I think these marsh organs are probably a pretty good model of that.
Kathy Todd Brown, do you want to ask a question?
Sure. Great presentation, Jim. Thank you so much. So one of the things that I was wondering was you had a really cool graphic on fertilized and unfertilized behavior between the the different nutrients and the biomass. And I was wondering what the implication of that was for the MEMS curves that you were presenting earlier with the stability points. Would those curves still be applicable in a system that was maybe nutrient polluted? So if you alleviate the nutrient limitation, would you still expect those types of that bell curve that you so beautifully demonstrated there?
Well, you mentioned nutrient polluted. If there is a site that is hypereutrophic to the point where the nutrients are actually a detriment to plant growth, then I don't know. I don't know if the curve would still apply or not. I do know that they apply in situations where the plants are nitrogen limited.
Okay.
Like they are in Plum Island, for example. All right. Now, Plum Island is the birthplace of this controversy about nutrient effects. And I'm not buying that, because I've worked in that marsh for many, many years, and including making a lot of SET measurements in that marsh and fertilized and controls. And we don't see negative impacts of fertilizing in Plum Island. You know, we can talk about I can talk about what I think was wrong with that experiment, but probably offline.
Sounds good.
I can stop the recording later, but maybe we have time for one more question. Dr. Clark, Mark, if you'd like to ask one more.
Thank you, David. Jim, excellent talk. Really amazing duration of work. It really, you can't even address some of the questions without that sort of extension of work. My question is related to partitioning, I guess, the particulate accretion component versus the plant accretion component. And I realize they're synergistic. But as you move into those deeper waters, you might have increased particulate, not just to stimulate plant growth, but maybe you've got more opportunity for particulate accretion. But also as you move along an energy gradient where wave energy may disrupt finer particulate accretion, whereas high marsh may be different, although it may be less available. I'm just wondering how you tease those apart, or didn't you, because they are synergistic?
Yeah, that's another good question. So with the work I've done in North Inlet to date, I really cannot separate the two effects, the effect of the plants, the increased growth on mineral sedimentation versus biovolume growth, because they are, they're correlated. Okay, so you've got an autocorrelation problem. And so I'd have to tease that apart experimentally, which I can do. But I think the other part of your question about waves and so on, I mean, you notice that I did not mention erosion. Okay, so MEM does not address erosion. And so if you have you know, especially erosion along creek banks and so on, I cannot model that with the current version of MEM. I would go, in that case, I would look at more, you know, geomorphic principles, like the relationship between tidal prism and the volume of the tidal prism and the volumes of the creeks. Okay, so we know that, you know, creek volumes increase to accommodate increasing tidal prism. So, you know, there are ways to do that, but not through MEM. MEM is really more of a model of the platform itself, and not, you know, I stay away from creek banks. Although there is a model, there's a hydro-MEM model. MEM has been linked to the model AdCirc. And that's a pretty cool model. And that does indirectly allow for creek expansion and related things. And what's interesting really about that is that if you think of a marsh landscape where the tidal amplitude varies spatially over this landscape. Well, that affects the way the plants respond to rising sea level, which also then affects the hydrodynamics. So there's this really complicated feedback between those two things, and it plays out over this, you know, three-dimensional landscape. And it, you know, it makes my head explode, but it's really cool to see. You know, yeah. So that was a good question. Thanks.
Thank you.
Well, thanks everyone for attending today, and thank you, Dr. Morse, again for the presentation. We really appreciate it. Good to meet you in close to real life. It was an honor.
Likewise.
Thanks everyone. So we'll see you all next week. Yeah, please do turn in your summaries. All the students, you have some latitude, but might as well do it now. Thanks, folks. Cheers and happy day.
Yeah.
Cheers.